Welcome everyone to today's edition of the EV Journal Club. We're very happy to have you here today. And we're also very happy to have a speaker who will be presenting some of her work on membrane binding peptides and how we can use them for extracellular vesicle capture and also analysis. So as most of you know, single particle analysis, single EV analysis is a real frontier in the field right now. And to understand the diversity of EVs, the diversity of their function, we really have to understand their makeup. And so capturing them for these single EV studies is extremely important. And then of course, characterization is the next step. So our speaker today is Marina Cretich of the National Research Council of Italy. And so she's working in Milan, and she has just recently published a paper with some co-authors in the Journal of Extracellular Vesicles, which, as you know, is the mouthpiece of the International Society for Extracellular Vesicles. And she's going to be presenting some of that work today, and perhaps a few other things as well. So Marina, thank you so much for joining us. And I would just like to note that what we're going to do today is leave the chat box open for people to ask any questions or make any comments for you. And I will try to compile those into something coherent so that we can have a good discussion at the end. So Marina, I'm going to hand it over to you, and thanks again for joining us today.
Okay, thank you very much, Ken, for your kind introduction. I will start by sharing my screen. Tell me if it works.
Looks good.
Okay, great. So thank you again, Ken, for the introduction and actually for inviting me today. This is a great opportunity for me and for the group. And actually, congratulations to you and your group on, I would say, starting and running these EV Journal Club events that are becoming more and more important for the EV community. So thanks again. So as Ken mentioned, so first of all, good morning or good afternoon, everybody. And as Ken mentioned today, I will present you our recent work on membrane binding peptides for small extracellular vesicles on chip analysis. And this is the overview of my presentation today. I will start by briefly introducing EV microarrays that are microarrays dedicated to the detection of extracellular vesicles. I will show you how we used membrane curvature sensing peptides to build our EV microarrays. Then I will show you some insights into what we speculate it's the binding mechanism between these peptides and the captured vesicles. I will spend a few words on the role of microarray surface chemistry, showing you why it is important, this aspect, why it's actually crucial for the success of the experiments. Then I will show you our data on the size dependency of EV capturing, and I will end with some conclusions and perspectives on the ongoing work in our lab in these days. So basically the context of this presentation is EV microarrays that, as you might know, were introduced by Malene Jørgensen and collaborators a few years ago. They are based on the use of antibodies that are immobilized on the surface of a microarray and used to capture extracellular vesicles on the basis of specific proteins that the vesicles have on their surface. And typically, there is a detection step following the capturing step in which it is added a cocktail of fluorescent antibodies, typically anti-tetraspanins antibodies such as CD81, CD63, CD9. And this, in order to enhance the detection of the vesicles that are captured by the antibodies. And this tool, this analytical tool, the EV microarray, is actually very helpful to characterize, to phenotype the types of the proteins that are on the surface of EVs. But today I will introduce peptide microarrays in which we do not use antibodies to capture EVs. Instead, we use peptides that are immobilized on the surface and used to capture the vesicles. And we can have at least two kinds of mechanism of capturing. We can have peptides that has an affinity, specific affinity for surfaces, for proteins on the surface of EVs, and we can call them, in a sense, peptide aptamers. And there are examples of these mechanisms. For example, there are amino acidic sequences published of peptides that have affinity for CD63, for EpCAM, for HSP70. Usually, these probes are developed by phage display library screening. And then there is another mechanism, which is the one on which I will focus today. That is the mechanism of membrane sensing peptides. Peptides are able to, let's say, to capture vesicles only on the base of the curvature of the membranes of EVs, thus being thus being able to capture the vesicles unbiased by the differential protein expression on the surface of the vesicles. And okay, in fact, the membrane of small vesicles can be considered a general yet specific marker in the extracellular space, because the membrane of small EV is characterized by a high curvature. Actually, the smaller the vesicles, the higher the curvature. And there is also a relative abundance of unsaturated anionic phospholipids in the external leaflets, typically, for example, phosphatidylserine. And also we have lipid packing defects, kind of holes floating on the surface of vesicles due to the high curvature of small EVs. And there are many biological processes, including vesicle secretion, that are specifically directed, I would say, or are organized by proteins that are able to sense the curvature of vesicles with great sensitivity and specificity. And so peptides derived from such proteins can be used as probe for small vesicles. And the mechanism by which these proteins act are very, I would say, complex and not yet well elucidated in detail in many cases. One of the most studied phenomenon or mechanism is the one of amphipathic peptides. These are peptides that first approach the membrane of EVs by an electrostatic interaction, which is the driving force of the interaction. Then these peptides are able to fold into the membranes by inserting their hydrophobic groups, the hydrophobic groups of their hydrophobic residues, into the membranes to stabilize the binding between the peptide and the membrane. But this is just one of the mechanisms. Actually, there are many different mechanisms. And I suggest you to have a look at this great talk that Professor Hubert gave a couple of years ago. And in this talk, Professor In showed many of these mechanisms. And also, I'd like to underline that Professor In is the first that introduced the rational design of peptides, of membrane sensing peptides, to sense the curvature of vesicles, of lipid nanovesicles and extracellular vesicles. So anyway, based on these rationales, and actually starting from one of the papers from Professor In that published in 2013, a bradykinin-derived peptide able to label in solution small lipid nanovesicles. So starting from this sequence, we have designed molecules, probes, engineered peptides, starting from this 9 amino acidic sequence that is here reported. We call it bradykinin-derived peptide, BP, and we also designed a mutated peptide in which the arginine residues were mutated to glutamic acid to revert the electrostatic charge of the peptide from positive to negative. And this is our negative control. And as you can see, both molecules, both probes bears a spacer and a linker, a PEG linker, a short PEG linker, and a propargyl glycine, which is a non-natural amino acid that we use to bind the peptide with high chemoselectivity to the surface of microarrays by the use of a polymeric coating. by a click chemistry reaction. Basically, this polymer coating is able to provide on the surface of microarrays and azido groups, this one, that can react with high selectivity with the alkynes of the propyl glycine. So what we do from the practical point of view is to prepare clickable, what we call clickable chips that are silicon slides coated by the functional polymer bearing azido groups that are exposed on the surface in such a way that when we spot the peptides bearing the propyl glycine, the triple bond, the alkyne group of the glycine is able to react with his orthogonal group on the surface by a well-known click chemistry reaction, which is the copper-catalyzed azido-alkyne cycloaddition. So in this way, we are sure that our peptide is immobilized chemoselective in an oriented way. And we have also designed peptides, probes, in which this bradykinin-derived sequence, the BP linear sequence, is repeated twice in a branched and tandem conformation to provide a multivalent representation of the peptides from the surface. And today in this presentation, I will show you data only about this peptide for the sake of brevity and clarity of this presentation, but you can find details on these multivalent presentations and the comparison of their performance in the JEF paper. And I'd like also to say that this concept of chemoselective oriented immobilization of peptides and multivalent presentations originates from our previous work on the development of peptide microarrays for serological applications, for the detection of antibodies in diagnostic applications. And we have, let's say, applied this concept, this platform to peptide microarrays for EV, for extracellular vesicles. Well, the detection platform that we use is the ExoView platform by NanoView, that, as you might know, is based on a technique called single particle interferometric reflectance imaging sensors, SP-IRIS. This is a label-free technique which is able to make use of these silicon chips that are spotted in the center with the spots of capturing probes, in our case of peptide probes. The instrument image each spot and is able to label-free detect, count, and size the single vesicles that are captured on the surface. And this technique was originally developed by Professor Selim Unlus and his group at Boston University. It is based on the interferometric enhancement due to this silicon oxide layer here on the silicon chip, interferometric enhancement of the scattering signature of the bio nanoparticle that is captured on the surface. Actually, this technique was originally developed for viruses, only lately adapted to the detection of extracellular vesicles. Anyway, with this technique, we are able to label-free count and size the captured particles. The company NanoView later implemented into the commercial platform a fluorescence detection. So the capacity to detect by fluorescence using specifically 3 colors of fluorescence proteins that are on the surface of EVs by using an immunostaining. So what we do from a practical point of view is to prepare our chips. That means we coat with our functional polymer the silicon chips. We spot the peptides. We know that they bind chemoselectively in an oriented way on the surface. And in this way, we produce our own peptide microarrays. Then the surface is incubated with a sample containing extracellular vesicles that are captured by the probes. The NanoView platform is able to label-free count and size by SP-IRIS technique the vesicles that are captured at each spot location. And then optionally, we can add fluorescent antibodies for a fluorescence colocalization of protein markers by immunostaining on the surface. And so we started from a sample of EVs isolated by ultracentrifugation from a hex cell line culture that, according to the MISEV 2018 recommendations were extensively characterized by transmission electron microscopy, by nanoparticle tracking analysis system, and by Western blotting to confirm the presence of external and luminal EV, typical EV markers. And then this sample was incubated on the array, on the peptide array. And this is the typical results of one of our experiments. This is the spot of bradykinin-derived peptide. And the blue dots here are the vesicles highlighted by the analysis software, of course. So the blue dots are captured vesicles on the spot of BP peptide. This is the negative control peptide. You can clearly see that the binding of vesicles here is negligible. And you can also appreciate the background around the spots, which is very, very clean. And this is the size distribution of captured vesicles provided by the NanoView instrument. As soon as we did these experiments, we noticed a preferential binding for the small vesicles, so from 50 down to 150 nanometers in size. However, we will go back to this data at the end of my presentation. Then we performed a dose response experiments in which we made serial dilutions of our sample from 10 to the 9 down to 10 to the 6 vesicles per ml. We incubated this sample on the peptide array, and the blue bar reports the number of vesicles captured by the VP peptide, whereas the velvet bar here reports the particles captured by the negative control. These numbers are expressed as kiloparticles per square millimeter. And you can see that looking at the dilutions that we could easily detect concentration of extracellular vesicles down to 10 to the 7 extracellular vesicles per ml. Whereas the same samples incubated on an antibody array, a chip bearing antibodies anti-CD81, CD63, and CD9, allowed us the possibility to detect only the highest concentration, the 10 to the 9. So this data, the comparison between these data suggests that the peptide array actually has a much higher capturing capacity if compared to what is captured by these antibodies on this array. And we ascribe this phenomenon, we speculate that this phenomenon is possibly due to a cooperative effect of multiple peptides acting simultaneously from the surface to capture and keep one of the vesicles on the surface of the chip. Whereas in the case of the antibody array, one antibody is able to find only its own marker. Well, then we also confirmed the EV nature of the vesicle, of the captured vesicles. And so we performed an on-spot staining of the chip using the anti-CD9, CD63, and CD81 antibodies, fluorescent, labeled in 3 different colors of fluorescence and imaged in 3 different fluorescent channels by the instrument. These are the vesicle, the CD81 positive vesicles captured on the spot, the CD9 positive vesicles, and the 63 positive vesicles captured on the spot. Again, you can see that the background around the spot is very clean, and also that the capture by the negative control is basically not detectable. So then we move to more complex samples, because this is samples of EVs isolated by ultracentrifugation from a hex cell line culture. We move to more complex samples, and we move to human serum and human plasma. We applied conventional isolation tools such as ultracentrifugation, size exclusion chromatography, a combination of size exclusion chromatography and polymer precipitation. And the results also from serum are very similar to the one that I've shown you. You can find all the details in the paper, of course. And then we move to challenge our chip with really a complex sample. We use untreated serum. So basically, we just diluted serum 1 to 10, and we filtered it by a 0.2 micron filter, and we incubated this on the surface of the chips. And in this case, surely the specificity of the capturing and the affinity is not as good as when you use a highly purified sample, and I think this is really normal, expected. However, what I'd like to underline here is that when we use the multivalent presentation of the peptides, so the branched or the tandem configuration that you can see here, the specificity and the affinity of our capture really increase, demonstrating a specific role of an important role of multivalent presentations, especially in complex samples. So right now we are working to improve our protocols with using multivalency to have better results in terms of specificity when we use highly complex samples such as pure serum or pure plasma. And then we wanted to have insights into the mechanism by which the membrane sensing peptides capture the vesicles. And what we did is an experiment of trypsin shaving or trypsin digestion of the proteins on the surface of extracellular vesicles. So we took a sample of EVs that were isolated from human serum by ultracentrifugation, and we subjected the sample to 2 protocols. We incubated at 37 degrees for 6 hours without or with trypsin. And as expected, on an antibody array where we have immobilized CD81, CD63, and CD9, the binding of the vesicles is abolished by the protein digestion. We do not have any binding due to trypsin digestion. This is expected. On the other hand, the same samples were incubated on the sensing peptide array, provided us no difference. I mean, the 2 samples not treated with trypsin or treated with trypsin provided us basically the same capturing, thus strongly suggesting that the binding is not influenced by protein digestion, and suggesting that the binding is not mediated by proteins and is probably due only to the lipidic components of the membrane of EVs. And then I'd like to spend a few words on the role of surface chemistry, and trying to convince you that this is a very important aspect for any microarray experiment, and specifically for this type of microarrays. Our lab has a long-lasting experience and expertise in developing polymeric coatings for microarrays. And here it is, this is the polymer that we call MCP2, is a polymer that had in the past and has actually a great success for proteins, antibodies, and for amino-modified oligonucleotides. Actually, this is my favorite polymer. And it is a tercopolymer made of the backbone of dimethylacrylamide, which is the major components of this polymer. We have a selanating moiety, and we have the functional group, which is a NAS, an ethylenoxysuccinimide, which is able to react randomly with any nucleophiles in the molecule that you are spotting on the surface. So basically, any amino group of your protein or your antibody or your peptide can react with the polymer and be immobilized covalently on the surface. And this polymer is great when we use proteins and antibodies. When we move to peptides that are very small molecules, very tiny molecules, it is important to have a chemoselective immobilization. So that's why we use this MCP6, which is a derivative of MCP2. The components of the polymer are exactly the same except for the functional monomer here that is not an AS, it bears an azido group, which, as I mentioned before, is able to react chemoselectively only with the bioorthogonal group that is the proper G glycine in the peptide that we spot on the surface, in such a way that we are sure that the peptide here is immobilized in an oriented way and it also spaced from the surface due to the linkers that we add during the synthesis. And the difference between these 2 surfaces, even if just a minor component change, is dramatic. You can see here what we capture, how many vesicles we can capture using this polymer, and how poor is the binding when use this other random polymer. And this is the difference if you look at the number of kiloparticles in the chemoselective versus random chemistry. So this is to underline the role of surface chemistry, how it is important to tailor, carefully tailor the surface when you use peptides that you have to imagine it's a very small molecules comparing to these big balls that are the vesicles that are captured and sitting on the surface. Well, and then I'd like to tell you something about the size dependency of the peptide capturing. And I'd like to tell you that when we submitted the paper the first time to JAV, the only data on size dependency of peptide capturing were the one that I showed you at the beginning with the size distribution of captured particles provided by the NanoView platform. Well, and I have to thank one of the anonymous reviewer of JAV that asked us to investigate more on this aspect. And I have to thank he or she because the reviewer suggested some interesting experiments that in the end to me added value to the manuscript. So the reviewer suggested to compare the binding of liposomes of the same lipidic compositions but different sizes. And so we purchased from a Spanish company these pro-nanosome lipo-N that are liposomes of a lipidic composition resembling the one of EVs, but of different sizes that were characterized by them and confirmed by us. And we wanted to compare the binding of these liposomes with the peptides, but we had the problem that the NanoView instrument have a limited detection range from 50 nanometers to just 200 nanometers in size in the large part of the size range. So in case we could have detected the larger liposome of 200, 250, and 450, we risk to not be able to see it. So we use another detection technique, which is surface plasmon resonance imaging. It is a surface plasmon resonance instrumentation detection platform coupled to imaging, SPRI. That means that it's a kind of a mixture between a microarrays, but with a surface plasmon resonance detector that can detect the accumulation of mass on each spot of a microarray, that can monitor online the accumulation of mass on each spot. And we are lucky because we have in place a collaboration with a group in Milano from Fondazione Don Carlo Gnocchi, and I thank Silvia Picciolini for running these experiments. They have published in Analytical Chemistry in 2018 a paper on the surface plasmon resonance detection of EVs on an antibody microarray spotted on the SPR chips that are these gold chips that you might have seen before. And we are also lucky because previously we have worked with this group to develop a surface chemistry to immobilize peptides on these gold chips for SPR imaging. So, and this chemistry is exactly the same that we use for the silicon chips that I've shown you a few slides ago. So basically, we could directly compare the results that we have on the silicon chips and the NanoView instrument with what we had on the gold chips with the SPRI instrument, the surface plasmon resonance instrument. So what we did was to develop the peptide microarray on the SPR chips, and when we injected the samples of the liposomes with different sizes, the results were very clear actually, because we had a good SPR signal. This is a sensogram, a very good SPR signal for the 150 nanometers liposomes. whereas we had basically no signals, so no accumulation of mass, no binding for the larger liposome, the 250 in orange and the 450 nanometers in gray. So, and these results are actually in perfect agreement with the original paper by Professor Yin on the sensing of small lipid lipovesicles with the bradykinin-derived peptides. But of course, this is just a model, and the reviewer suggested another interesting experiment. The reviewer suggested to compare the binding of two samples prepared by two different ultracentrifugation protocols. So we started from an unfiltered platelet-free human plasma, and we subjected the samples to 100 kg ultracentrifugation for 2 hours to obtain a small EV enriched sample. And we, to another kind of, another ultracentrifugation protocols, 10 kg for 1 hour to obtain a microvesicle enriched sample. And both samples were characterized by NTA and by Western blotting. And then these two samples were incubated on the peptide chip. And the results are the following. You see that the size distribution of these two samples is basically the same. It's very, very similar. It's basically the same, meaning with the preferential binding for the small vesicles from 50 to 100 or maybe 120 nanometers. This suggests that no matter if you have a small EV enriched sample or a microvesicles enriched sample, what the peptides on the surface captured are only the small EVs. And interestingly, this is not the case on an antibody, on the antibody chips. These are the antibody chips with CD81, CD63, CD9, and CD41 immobilized. And clearly the size distribution are larger, are wider, and especially here, it's interesting that the vesicles, the size distribution of vesicles captured on CD41, which is, as far as I read on papers, according to the literature, it's a marker related to platelet-derived microvesicles. So especially on these antibodies, the size distribution is much larger compared to what is captured on the membrane sensing peptides. So to summarize, I've shown you today the first example of peptide microarray applied to EVs. I've shown you as we can use peptides as alternatives to antibodies immobilized on the surface, and the advantages over antibodies are many. The advantages of peptides, it's low cost, high stability, no batch-to-batch variation, high synthetic versatility, which provide us many possibilities for functionalization. And this is very important for when you want to functionalize, bioconjugate your molecule and functionalize a surface. And specifically, I've shown you how to use the use of membrane sensing peptides in this application. We believe that these peptides can be considered universal ligands unbiased by the differential expression of proteins on the surface of EVs, so they can be coupled to antibodies for your specific markers, in which you are interested in. They can be coupled to them and used as possible reference and standardization tool, which is very important to have to enhance the reliability of our experiments. I've shown you that peptides have provided us a high capturing capacity, probably due to this intrinsic multivalency or cooperative binding of many peptides on the surface. And I've shown you that they are generic baits for both microvesicles and small EV within the overlapping size range of these samples, that is probably 50, 120 nanometers. And I've shown you that they can be applicable also to complex samples such as untreated serum or plasma. And so what's going on now in our lab? What are the new directions? We are working to enlarge the panel of membrane sensing peptides, introducing new sequences. We are working to develop peptidic probes for specific proteins, and we are now setting new collaborations to use phage display library screening for these. And we are trying to use these membrane sensing peptides as decorating agents for EV. In this case, we synthesize what we call hybrid probes, that are probes made of a part is a peptide with affinity for the membrane, and there is a linker with a reactive chemical handle able to react very selectively with a tag in a protein or in an antigen that you want to decorate the EV with. And we are also using this platform as isolation tools in experiments of capture and release. Here, the hybrid probes will have a cleavable linker in such a way that after capturing the EVs on a solid support, for example, a bead, upon the use of a proper stimulus that can be UV light, an enzyme, a redox trigger, the linker can be cleaved and captured vesicles can be released for the usage. And actually, this platform is at the base of our very recently funded European project, Marvel, in which we will develop a scalable and reversible tools for extracellular vesicles. This is a project that will start on November the 1st with 5 partners in Europe. And in Marvel, we will use the functional polymer coatings to functionalize different solid supports, bind the membrane sensing peptides and other specific probes to these solid supports by cleavable linkers, so that with a stimulus we can release and collect the vesicles that we have captured. And in Marvel, we aim to integrate this platform into different supports that are with many formats, such as beads, chromatographic columns, or tangential flow filtration membranes. And I know it's an ambitious project. It will be a long journey, a big adventure. So stay tuned, follow us on Twitter to stay updated with our results. And in the end, I really like to thank the group I belong to, the Chemistry and Technology for Bioscience group at CNR in Milano. I'd like to thank all the members of the group, but particularly Alessandro Gori, who is the peptide scientist, and Marcella Chiari, who is my mentor and my boss. And I'd like also to thank George and David from NanoView, Silvia from Fondazione Don Carlo Gnocchi, Riccardo and Silvia from Ospedale San Raffaele, Natasha Zarovny from ANSA Biomed Exosonics. A special thank to Evita, the Italian Society for Extracellular Vesicles, for continuous support to our community. These are our current funding. This is very important. Actually, I'd like to thank very much the European Project Index, which is coordinated by Marcella Chiari. This project actually introduced the concept of capture and release, and that we are applying within a complete system for on-chip analysis of exosomes. And the other project that we have now in place is Hydrogex. This is a regional project coordinated by Alessandro Gori. In this project, we will develop a peptide hydrogel platform for isolation and multimodal analysis. In the very, very end, I'd like to thank you very much for the kind attention, for the time you spent today to listen to me. I will be happy to take questions, of course, but should you have any feedback or wish to collaborate with us, please feel free to drop me an email at any time, and thank you very much.
Well, thank you, Marina. That was a wonderful presentation. I feel like I've learned a lot, and I understand a lot better what you've been able to accomplish, and then also what some of the promise is with this technology, or this set of technologies. So we have had some very good questions in the comment box, and there was one that I feel was stolen from me. I wanted to ask it, but I think it's on several people's minds. So we had Daniele and also Andres Zielstra. Some others had asked a question here. So Daniele, could you unmute yourself? Do you want to ask this question?
Yes, thank you for the presentations. It was very interesting. And I was wondering if you test the bond of lipoproteins or if it is specific only for extracellular vesicles.
Okay, thank you for this question. Actually, when we isolate EVs from human plasma and human serum by different isolation tools, we check that we didn't have any we check the ApoE marker to be sure that we get rid of many of the lipoproteins, and when we use these samples. Anyway, this is a very good question, a very important aspect. We are in the protocols that we are now developing. We wanted to do exactly this. We wanted to spike in lipoproteins in a isolated, in a very purified EVs to be sure, or to confirm that we didn't bind any lipoproteins.
Okay, thank you very much.
You're welcome.
So just to follow up on that, I mean, theoretically, though, you could have peptides that would bind specifically to lipoproteins. Is that right?
Could be.
And that would be, I think that would be interesting as well, because there is the proposal that that lipoproteins will also carry some of the cargo, like extracellular RNAs that EVs are well known for, but these might be alternative carriers. So that's certainly something that that a lot of us are interested in. So that's quite interesting to think about. And in fact, I'd like to hand this over now to Andres. Andres, you had a similar question, but then you also asked about the lipid composition. Do you want to expand on that?
Sure, thanks, Ken. So I asked
Andres, we can't hear you. Very faint. Okay, well, what I'll do is I will ask what Andres was asking. Or Andres, are you there now?
Yeah, can you hear me now?
Okay. Yeah, this is good.
Awesome. So I was thinking both about lipoproteins and the composition of other, the lipid composition of other EVs. In particular, we know that there's some cholesterol enrichment in some of the EVs and not in others. And I was wondering if there's differential binding of EVs based on their lipid composition, or if you had explored that.
We didn't have explored yet. Sorry, but this is very interesting suggestion, actually. Yes.
Yeah, it's because we know that there is a big difference in the lipid composition among the different EVs, particularly the smaller EVs. So that would be interesting to consider.
Yeah,
and just as an experiment that I would recommend to you, you can make liposomes of known lipid composition. So if you can make liposomes of fixed size but of differential cholesterol composition, for instance, then you can test that idea, because if the answer is no, then it doesn't really matter and you don't have to figure it out in the EVs.
Yeah, that's a very good suggestion. Thank you. Sure.
Yeah, I'm fascinated by that too, because one thing that we'd like to be able to do is to bind any EV universally. So that's one thing that we could do. But then on the other hand, if somehow function is related to that lipid composition, then you could also look for subtypes that way, and looking at the lipids rather than the just the surface protein. So it's a lot of possibilities there. We also had, so let me go to a set of questions that we had that was related to size. So to some extent, I think you addressed this after the question was asked, but I want to come to Corey. Corey, you had asked about the affinity and the affinity in different size ranges. Do you want to talk with Marina about this a little bit?
Yeah, I had originally asked this right before she started going into the different size ranges with the ordered liposomes. So I really think she honestly already addressed my question, but if she has any further elucidation she could provide in terms of the size ranges, and if that was also found in biological samples, as opposed to also the commercially made liposomes. I'd love to hear a little bit about that. Thank you so much for a great presentation, by the way.
Thank you. Yes, actually the results with the liposomes were very, very clear. We didn't expect such a clear response from the surface plasmon resonance, but also we were surprised by the real samples of EVs, not the model sample, but EVs purely isolated by different ultracentrifugation protocols. And that results are very clear in suggesting that what we capture is just, it depends on the size. So probably not so much on the different lipid composition or of course, the biogenesis that we cannot demonstrate anyway, but the fact that we can isolate the overlapping size range of both small EVs and probably microvesicles demonstrate that the sensitivity, the specificity for the curvature is really high. And also this was, I think I mentioned it, but this is in perfect agreement with the original paper. In the original paper by Professor Rin, he tried to label and to detect by fluorescence liposomes of different sizes, and he is the one that first stated this small EV affinity for the bradykinin-derived peptide.
Good.
Thank you so much.
You're welcome.
And you know, related to that question is one that came in from Randy Carney. Randy, are you there? Would you like to address this?
Yeah, thanks, Marina. Really nice talk. I really enjoyed that. It's fun to see the peptide stuff because I think it's really important and different from what other people are doing. So I had a question because you know, we always care about the size with EVs, and it's really apparent that they're not they don't have a mode at 100 nanometer like we're classically thinking with NTA and other type of light scattering. But still, you're limited by the refractive index of the particles, even in view down, and they, you know, are capping their size distribution at 50 nanometer. Probably they're smaller ones. I noticed some EM images that you took. Did you look for these smaller ones on chip afterwards, or do you have any comment on to what extent those 30 to 50 nanometer ones are present or variable in the samples?
Yeah, unfortunately, we are limited by the instrumentation, by the size range of the instrumentation. So I cannot exclude the binding of even smaller vesicles, maybe 40, 30 nanometers, probably. But we are not able to see them so far.
Yeah, that's, you know, when we use the ExoView instrument and we use the SP Iris mode, we might get, you know, 3,000 part events binding to a spot, but then when we switch to fluorescence for the, you know, the detection of other surface antigens, we might have higher numbers than you know, what we could detect on the spot, and that means that there is this population of smaller particles that you might be able to detect with, you know, with fluorescence that are not necessarily measured with the SPI.
That's correct, right?
So there's definitely a different another population there, and Mike Pilatus, you also had a question about how these sizes were being measured.
First of all, very nice talk. I really learned a lot from it, but I think it was answered with the last question. I really wanted to know how low in size these peptides could actually capture the EVs. 50 was the cutoff. I assume that was from the ExoView instrument. I assume you'd see them on EMs though, if you captured them on your chip, or if you could tag your peptides, you might be able to see the peptides attached to the vesicles on EM.
Exactly. In fact, one of the experiments that we would like to run in the future is to use either AFM, atomic force microscopy, or TEM, and with labeling to see which is the limit in size in small size. But anyway, what Ken mentioned before about the fact that with fluorescence on the NanoView instrument, we can detect also the smallest particles. This is something really happening a lot to us. We do very often see many more fluorescent particles with fluorescence than what we see level 3. And this suggests really that we can go down from 50 to 30 nanometers.
Randy, did you have a follow-up to that? Okay, great. All right, thank you. So the another question that we had to do with your treatment with trypsin. So Safia, are you there? Would you like to ask a question?
Yeah, thank you for a very good presentation. What I'm wondering is, I know that trypsin can sometimes interfere with the permeability of membranes. So I was wondering if your peptides were able to distinguish between membranes that are still intact, or does it just indiscriminately attach to any sort of membrane?
Okay, actually, the NanoView instrument detect the intact vesicles, that one that has a round shape, not the open membranes. So what we count them is probably only related to the small vesicles. And I'd like also to mention that the protocol that we use for trypsin shaving was actually the one that has very recently published Professor Yang-Song Go about the proteome, the real proteome of vesicles. And I remember that the ISEV in Kyoto last year, he presented in a poster this protocol, and I actually was inspired by this protocol, and he showed some very beautiful TM picture of intact vesicles after trypsin treatment, and we use the same trypsin treatment that he used in that work.
All right, thank you.
Great. All right. So so we also had a question from Benedetta Bussolati. Benedetta, are you there?
Yeah. Hi.
Hi.
Hi, Marina. So it's really fascinating what you're doing. So we are really keen to start using your kits. And so my question, I saw you have a dose response, and you had nice results starting from 10 to the 7 or 10 to the 8 vesicle. I don't know if it was 4 ml or 4 microliter.
Per ml.
So theoretically, it should be compatible with the number of vesicles that can be found in serum or in urine without isolation. So what do you think about that?
Yes, exactly. In fact, what we do with serum is to dilute the serum 1 to 10, filter and apply to the chip, and we have a good signal.
And also with cell culture medium?
With cell culture medium, yes. We in this case actually we are at the limit. Without any isolation from cell culture, in 3 days of culture, we are at the limit, honestly.
Okay. Thank you.
Good. So I think we have 2 more questions that we'll get to today. Madeline Longjohn, you had a question about aggregates. Madeline, are you still here?
Yes, I'm here. Hello.
Hello.
Hello. Thank you very much for your presentation. My question, I have 2 questions. The first one was to find out if you guys checked if this chip can be used to potentially capture EVs from serum and then immunophenotype them. And then my second question is, I may have missed this, but just to be sure, does the chip differentiate EV clones and EVs that are in complex proteins or even protein aggregates?
So the first question is, yes, we can use these chips with complex samples such as serum and plasma, yes. And now I forgot your second question, sorry, can you repeat?
Okay, when I said, I meant immunophenotype them, so capture the EVs and then find out, also state to find out the proteins both on the surface and in the lumen of the EVs.
The luminal EV, okay, the luminal. We didn't do this now in the lab, but I know that NanoView company has protocols to do this.
Okay.
I know they have.
Okay. Okay. My second question is if the chip can differentiate between EVs, like single EVs, or EVs in clumps, or EVs in complex with proteins, or even protein aggregates.
Okay, this is a good question actually, but this is anyway, when you use fluorescence in the NanoView instrument, it is a sandwich experiment. So basically we have a double check because there is the specificity of what you capture using an antibody and a peptide, and it is what you detect. So the fact that you detect on a single particle that is intact, a tetraspanin in a sandwich mode means that you can be quite sure that you're capturing an intact vesicles and not a protein aggregate.
Okay.
Especially if you use the peptide, which is directed against the membrane, and then you detect the protein. And also, we have to consider that the instrument is able to size and to count the size the single vesicles captured on the chip.
Okay. Okay. All right. Thank you. Thank you very much.
You're welcome.
Yes, so the last question for today was actually the first question that came in, but I thought it was perfect to put it at the very end. So G. Bart, would you like to ask your question?
Yeah, can you hear me?
Yes.
Yes.
Oh, sorry. Yeah, I'm sorry. Yeah, my name is Genevieve. My office computer use my email address. I'm actually interested to know if you think that if NanoView is actually planning to produce and sell a chip designed with your method, because I think there's a lot of application, particularly people who work with non-human or non-mouse EVs, or do you know anything like that?
Yes, thank you for your question. Actually, we are in contact with them. We are collaborating. We know them since a long time, but you have to ask them. I have no connection with them. I'm not aware of their policies. And so I know that they are well aware of the technology. So maybe in the future, who knows, they will introduce it. I hope so, really.
Yeah, because that's really extremely high potential, I would say. But thank you.
Yeah, that's an extremely good point. That we have markers that have been developed quite well for mammalian EVs, but for many other sources, we just don't have the tools right now. And it would be wonderful if people could use a peptide like this as sensing a membrane rather than a specific surface marker. So I think that there's a potential for a real game-changing force here. So that brings us to the end of our hour today. I want to thank you all for joining. Thanks to everybody for your great questions. And most of all, thanks to you, Marina, and thanks to your great team that you put together there in Italy to bring us this technology, evaluate this technology. And I look forward to all of the advances that are going to be made as a result of your hard work.
Okay, thank you.
Thank you
very much.
And have a great rest of the week, and look forward to seeing you all soon. Take care now.
Thank everyone. Thanks.